All right, I want to spend a little bit of time now talking about membrane transport. In the previous lecture, we introduced some just basic concepts about the cell itself, the size of the cell, and then we jumped into a discussion about the plasma membrane and the fluid mosaic model, the structure and fluidity of the plasma membrane. And we said that one of the major functions of the plasma membrane of a cell is that it controls the interactions of a cell with its environment. All right. And when we say that, what we're trying to say is that a cell basically controls the movement of substances in or out of the cell. It can prevent, it can allow certain things to move in or out. Okay. And what I really want to talk about are basically diffusion and osmosis. The two major types of movement through a cell membrane. All right. Now, when we talk about these topics, we have to talk about a concept called selective permeability. Before I get into the concepts, we have to get acquainted with some terminology first. Okay, when we say that a plasma membrane is selectively permeable, what we're saying here is that it can allow some substances to move, through the cell membrane, and some substances may not move through the cell membrane. Okay, so for example, remember in the previous talk, so let's say there's a cell membrane, and let's say here is a sodium gate. Okay, and remember before I said that only sodium is going to pass through a sodium gate. That's it. All right, that's it. And, let's say there's a bunch of potassium here, and let's say there's some potassium here. Okay. Potassium is not going to travel through here. Okay, so if all we have are sodium gates, all right, potassium will not be able to cross or move through the cell membrane. All right, that's what I mean when I say selective permeability. All right, that cells can control what can move into or out of the cell. All right, and as you're already starting to deduce after hearing this, is that cells control their permeability to specific ions or molecules by controlling the amount of and the types of gates that are found within their receptors. All right, so keep that in mind. Selective permeability. Cells can select for what can move into or out of the cell, or how easily something can. So for example, if I have a ton of sodium gates on here, all right, but I only have one potassium gate, it's going to be a little more difficult for potassium to diffuse into the cell. All right, so kind of keep that in mind as well. That's selective permeability. So the plasma membrane is selectively permeable. All right, and there's two other terms you need to be acquainted with when it comes to understanding membrane transport, and that's passive versus active transport. All right, when we say passive transport, we're saying that basically there's no energy required. There's just a concentration difference that's allowing movement to take place. All right. And when we're talking about active transport, this is an energy requiring process. All right, this is moving solutes across the cell membrane via active transport requires the expenditure of ATP. All right, so let's kind of jump back here and talk a little more about this. Another concept that you need to know when it comes to understanding the transport across the cell membrane, and that's what we call the concentration gradient. Concentration gradient. All right. So basically, what a concentration gradient is, it's a difference in the concentration of a molecule or a solute or a particle. Well, let's call them particles across the cell membrane. Okay. So let's say we've got a cell here. All right. We've got a cell here. Okay, and let's say on the inside of this cell, I've got 10 sodium ions. Okay, so there's 10 sodiums. Count them out. You guys can use your imagination on that. And let's say on the outside of this cell, there are 10 sodium ions. All right, there are 10 sodium ions. Now let's say we put in a sodium-specific gate here. All right, so Now you have to remember when particles are moving across the cell membrane, all right, particles are going to be moving in both directions. Okay, particles are going to be moving in both directions. Okay, now what we have to talk about is there a net movement across the cell membrane? Okay, so if there are 10 sodium ions, within the intracellular fluid, and there are 10 sodium ions in the extracellular fluid, will there be a net movement? Meaning, will there be a real numerical change between the sodium in the extracellular fluid versus the intracellular fluid? And the answer is no, okay, because there's an equilibrium of sodium on both sides. There's an equal amount. Okay, so there will be sodium moving one direction, there will be sodium moving the other, and the concentration will remain the same. Okay. So that's not a concentration gradient. All right, if we want to illustrate a concentration gradient, okay, we would have to, let's draw this over again. Now what we would have to do then is say, okay, so intracellular fluid, you already know this is extracellular fluid. Okay, so now let's say there are 10 sodium ions on the inside of the cell, and now let's say there's 145 sodium ions on the outside of the cell. Now we have developed a concentration gradient. There is a net difference of sodium in the intracellular versus the extracellular fluid. Okay, and so basically what we're saying here, when we create a concentration gradient, what this is, it's a concentration difference across the cell membrane. Okay, so now there's a different amount of sodium on the outside versus the inside. Okay, so now as a result, there will be, transport taking place. So let's say I open this, again, this doesn't happen this way, but for the sake of discussion, let's say this sodium gate just opens. Okay, let's say this gate just opens indefinitely, and we leave it open. Okay, so the question you should be asking yourself then is, where will there be a higher net movement of sodium? Because like I said, remember, there will be some sodium diffusing out, some diffusing in, but where will you see a higher volume of movement? Okay, sodium diffusing in or sodium diffusing out? Okay, let's just say that this is passive. There's no energy required. We're just going to let the laws of nature do what they do here. Okay, what's going to happen is there's going to be a net movement of sodium into the cell. So there will be more sodium, far more sodium moving in than out. Okay. And if we just left this gate open, okay, if we left this gate open, sodium would continue to, we would have more sodium diffusing in, and this would happen until we reached an equilibrium between the extracellular fluid and the intracellular fluid. Okay, probably should have kept the math even here. Let's say that's 140. So now we've got 150. So now there would be 75 and 75. Okay, so movement will stop. Okay, when there's enough sodium movement, when we reach an equilibrium on both sides. Okay, so what happened here is sodium diffused down its concentration gradient. Okay, sodium diffused down its concentration gradient. Okay. So when I say the word diffusion, what I'm talking about is the movement of a particle across the cell membrane, like a sodium ion, a glucose molecule, a potassium ion, an amino acid, and so on. Okay? Osmosis is the movement of water across the cell membrane. So right now we're focusing on diffusion. All right? So when we're talking about passive diffusion, passive movement. Okay, what's going on here is if there's a difference in a concentration gradient, and there is permeability to this specific ion or particle, and what's going to happen is this ion or particle is going to diffuse down its concentration gradient, and diffusion is going to occur until one of two things happen. One, until we reach an equilibrium amongst both sides of the cell membrane, which pretty much never happens. Or two, the most common example is if this gate were to close, okay, because now if these sodium gates close, now sodium cannot, sodium cannot move or diffuse into the cell anymore. All right. So that's that's essentially passive transport. The movement of, the movement down the concentration gradient. Okay. And like I said, all a concentration gradient is a different amount of a particle on one side of the cell membrane versus the other. All right. So active transport would be about the exact opposite of this. All right. If we're talking about active transport, all right, let's say there are 10 sodium ions on the inside and there are 50 sodium ions on the outside. Okay, what's going to happen now is we do have some pumps that work against the concentration gradient, because normally, remember, normally particles diffuse down their concentration gradient, but there are certain situations when we need to work against it. Like, for example, when a nerve sends an electrical impulse, what we call an action potential, or what people commonly call them nervous impulses, okay, what happens is there's normally a higher amount of sodium on the outside of the cell versus the inside, okay, and when the nerve generates an action potential, it does this, it creates an electrical potential by allowing sodium to diffuse down into the cell. Okay, but in order to get that, to reset that cell, to get it back to its normal resting membrane potential, all right, we have to get the sodium back out. This is all going to make sense, and a little more sense a little later on when I talk specifically about this process. Okay, but so in order to do that, we need to get that little bit of extra sodium that came in back out, okay? So we're going to pump it against the concentration gradient back out of the cell. And in order to do that, you need to burn energy to do so, okay, or to do this process, all right? So that's active transport, moving against the concentration gradient, moving up the hill, okay, working our way up the hill. All right, so those are some basic terms you need to understand about membrane transport. You need to understand selective permeability. You need to understand what a concentration gradient is, and you need to understand the difference between passive and active transport. If you understand the basic concepts that revolve around these terms, okay, this will all make a lot more sense to you. So make sure that you're comfortable with these terms before you continue into this talk or into this topic. All right. Now, bear in mind, when we're talking about the diffusion of particles through the plasma membrane, remember that the plasma membrane, the chemistry of the cell membrane alone makes it selectively permeable. Okay, because remember, the plasma membrane is primarily composed of those amphiphilic phospholipids. Okay, with the polar heads and the non-polar tails. Okay, so yes, even though, remember, anything that is polar cannot just freely cross the cell membrane, okay, because it can't get through these the non-polar middle aspect of the cell membrane itself. Okay, so the membrane alone is selectively permeable. All right, but bear in mind, non-polar charged particles can, like I said, eicosanoids or lipid, steroid hormones that are derived from lipids. Okay, so for the most part, diffusion takes place through the cell membrane. Through proteins within the cell membrane, through those gates that I talked about in the previous video. Now, one thing that we do have to talk about, though, are the diffusion of gases. Okay, gases travel freely across the plasma membrane. Okay, so for example, let's say we have a cell here, and let's say there's a concentration, let's say there are 20 oxygens outside of this cell, all right, and then let's say there are 10 on the inside. Okay, we have created, there is a concentration gradient here, so these gases will diffuse right through the cell membrane into the cell, and the cell's going to burn that up for fuel. Okay, and then vice versa. With carbon dioxide, let's say there's, 40 CO2 on the inside and 10 CO2 on the outside, carbon dioxide will freely travel right out of the cell and eventually into the bloodstream. Okay, and you're going to learn about this when we get to the respiratory system and we talk about partial pressures of specific gases. What partial pressures are, all they are, they're just different concentrations of gases on, you know, in the extracellular fluid versus the intracellular fluid. Okay, so gases just freely diffuse right through the cell membrane. Okay, so when we talk about carbon dioxide and oxygen, they're not moving through channels or gates. Okay, they're just passing right through the membrane itself. Okay, so keep that in mind, and like I said, we'll talk more about this in this concept in A&P2 when we talk about the respiratory system and respiratory physiology. All right. All right, so on that note, let's talk a little bit about some factors that affect the rate of diffusion, because, you know, diffusion is going to happen, you know, a little more quickly in certain situations than others. I apologize, this slide looks really messy. When I uploaded this PowerPoint that I made into this program that I use called Notebook, some of the figures didn't transfer in, so I tried to fill it in with the pen here. So my apologies on that. So there are some certain factors that will affect the rate of diffusion, will affect the speed, you know, that particles will travel across the cell membrane. Okay, one of them is temperature. Okay, so basically, as we increase temperature, we increase the rate of diffusion. Because basically, remember, when you're measuring temperature, you're measuring molecular motion. You're measuring kinetic energy at the molecular level. Okay, so the higher we increase temperature, all right, the more molecular motion is going to take place. Okay, because you have to remember that when we're talking about this stuff, when we're talking about diffusion, you know, about, you know, particles moving across the cell membrane, they don't all just line up nicely and move right through the membrane or right through a gate when we want them to. Okay, you have to remember that these molecules, these particles, these ions are always in motion. They're moving around all the time. Okay, and basically diffusion is going to occur when these particles are moving around, and then they come in contact with the gate and then pass through it. Okay, if we increase the temperature of this environment, okay, we increase molecular motion and we increase the chances of this specific particle coming in contact with that gate and passing through it. Okay, so that's how temperature affects diffusion rates. All right, and so obviously the opposite would happen if we decrease temperature. Okay, there would be less kinetic energy at the molecular level, and it would take longer for these particles to diffuse across the cell membrane if temperature went down. Okay, so one, increasing temperature will increase the rate of diffusion. Okay, molecular weight. The smaller the molecule, the quicker it's going to get across the cell membrane. Okay, now I know you guys are probably thinking about this like, if I was on top of a hill and I took a bowling ball and rolled it down, and at the same time I let a ping pong ball go down. Okay. This doesn't work this way because you have to think in 3 dimensions. Okay, you have to remember cells are 3-dimensional objects found in 3-dimensional environments. So you have a cell here sitting in an environment. There's, you know, particles surrounding it all over the place. Okay, and basically the smaller the particle, okay, the easier it's going to pass through these specific gates to get into the cell. The bigger the particle, the slower and longer it's going to take to get in. All right, so it's not as simple as I'm just dropping something down from a high place, and the more dense, heavier object is going to work its way down. Okay, so the smaller the molecule, the quicker it's going to diffuse. Okay, just because it's gonna squeeze into the cell a lot more easily. Now next is something that we call the steepness of the concentration gradient. Man, this technology is awesome. Okay, the steepness of the concentration gradient. Okay, well, remember when we're talking about something like concentration gradient, what we're talking about, again, is just a difference in number across the cell membrane. Okay, so let's say we have a cell here. Okay, let's say we have a cell here, and there are 40 sodium ions on the inside. I know I'm throwing around a lot of random numbers at you, but just work with me here. Okay, there are 40 sodium ions on the inside, and let's say there are 60 sodium ions on the on the outside, 40 on the inside, excuse me. Let's say we take another cell and we have 10 sodium ions on the inside and 145 on the outside. Okay, now you should be asking yourself, which one has a steeper concentration gradient? Okay, remember what a concentration gradient is. It's just a difference in the amount of a particle across the cell membrane. A difference in the amount versus the intracellular and extracellular fluid. Okay, so this bottom one here would have the steeper concentration gradient because there is a greater difference in concentration. Okay, there are a lot more sodium ions in the outside versus the inside in this cell. Okay, whereas there's a difference of 20 here, all right, and there's a difference of 135 here. Okay, so if I open sodium gates within these cells, okay, even though even though there's only a couple on here, diffusion is going to occur much more rapidly because there's a greater difference. There's more opportunity for more sodium ions to come in contact with these gates and pass through them into the cell. Okay. So the steepness of the concentration gradient. Okay, the steeper the concentration gradient, the higher the rate of diffusion. Okay. The higher the rate of diffusion. You give the example of the oxygen mask here. That's why you flood somebody with oxygen if they're having respiratory issues or, respiratory complications, because, if you flood their system with oxygen in certain situations, okay, you're going to increase the steepness of, because remember, we said that gases freely diffuse right through cell membranes, right? Okay, so if we're having a respiratory disorder, whatever it may be, and we need to ensure that person gets oxygen, okay, you're going to flood them with oxygen, so more oxygen can get into the cell more readily and we can keep the cells alive. All right. Membrane permeability. Okay, membrane permeability is a big factor that affects the rate of diffusion. Is this the same slide? Yeah, it is. I just noticed that. Okay. Membrane permeability. Okay, the bigger the surface area, the higher the rate. Okay, because you have to remember that these cells, one more room over here. Okay, that these cells, again, are three-dimensional objects that we find in, within us. Okay, so basically, if we have a small cell here, if we have a small cell here, okay, and we have a big cell, all right. Remember, all this is remember this molecular motion and all this stuff. All this is about are, okay, so let's say we've got 10 sodium ions on the inside, 60 on the outside, 10 on the inside, 60 on the outside, an equal amount, okay? But what's going to happen here is it's a lot easier for molecules to disperse and get into the cell, the bigger the surface area. Okay, whereas with this smaller cell, okay, these molecules you're going to be randomly moving around, but they're going to be banging into each other and stacking up, and then it's going to be a lot harder for them to get into this smaller cell. Okay, so the bigger the cell, the more rapid the rate of diffusion as well. All right. Membrane permeability. Obviously, if we increase permeability, we increase the rate of diffusion. And as I mentioned before, cells increase their permeability by adjusting the amount of gates that they have. Okay, if a cell, so let's say here's a cell, and this is a sodium gate. Okay, if this cell needs, for whatever reason it may be, needs to make itself more permeable to sodium, it's going to produce more sodium gates. Okay. So the more gates, the more permeable, plain and simple. All right, so these are some very important factors that affect diffusion rates, okay? And that's why we strive very hard on a day-to-day basis. Remember, we talked about that concept of homeostasis in Chapter 1 to maintain a normal body temperature, okay? To maintain, normal concentrations of water, to maintain all these basic aspects of our physiology, because they will affect, what we're talking about right now. Okay, so keep that all in mind as we're moving through here. All right, and then we want to talk about specific types of transport across the cell membrane. All right, there's what's called simple diffusion, facilitated diffusion, filtration, osmosis, and then a couple forms of active transport. All right. Now, there's various different types of active transport. For the sake of what you guys have to know for my class, we don't really need to go terribly in depth with all the different types of active transport that can occur within us. But bear in mind, there's more than what I'm talking about. Okay. We could actually classify this vesicular transport as bulk transport. All right, and I'll talk more about that in a little while. Okay, so on that note, let's kind of work our way into, so remember, these here are passive. Okay, remember, when I say passive, these don't, these do not require energy for movement. All right. So let's continue with our discussion. So simple diffusion. Simple diffusion is the simplest one, coincidentally, the simplest one to talk about as well. And I've already been talking about it this entire time. So if I have a cell and I have a net difference in concentration, let's say I've got, 50 oxygens on the outside and 10 oxygens on the inside. Okay, with simple diffusion, basically we've established a concentration gradient. Okay, oxygen will, more oxygen will diffuse in, and I mean, that's all simple diffusion is just the basic movement down the concentration gradient because of a concentration difference. Okay, and earlier, as I mentioned, this is how gases move in and out of cells, just by basic simple diffusion. Okay, so you know that cells are constantly producing carbon dioxide as a waste, so there's always going to be more carbon dioxide inside the cell versus outside. Okay, because we eliminate carbon dioxide, we breathe it out. All right. So as a result, so being that there's more CO2 on the inside, there's always going to be CO2 diffusing out just by simple diffusion. Okay. And vice versa with oxygen. All right. And if you do have respiratory disorders, or you're having situations where you are accumulating too much CO2 in your system, this is going to affect this process. Okay, if you're unable to eliminate CO2 properly, if you're having certain metabolic disorders that are affecting pH and gas transport and all that, okay, this basic process will be greatly affected by this. All right, and like I said, I'll talk more about this when I get to the respiratory system much later on in the course, but that's all simple diffusion is. Just traveling down the concentration gradient. No energy required, plain and simple. All right. Now, the next one I want to talk about was facilitated diffusion. Now, remember that not all particles can just freely travel across the cell membrane like gases or nonpolar molecules can. Okay, because remember, there's a lot of particles out there that are polar. Okay, remember when we say polar, we're saying charged. All right, so since these charged particles cannot freely pass through the cell membrane, there has to be some way to facilitate the movement across the cell membrane, and that's where these gates come into play here. All right, so if we've got a plasma membrane here, and then we've got a sodium gate, it could be, it could be glucose, whatever it may be. All right, so let's say let's go with sodium. Okay, let's go with sodium. Actually, you know what? Let's talk about glucose instead. We're going to talk about sodium a lot in a little while, and you're going to get sick of it. So, like glucose, for example. Okay, glucose doesn't just freely move across cell membranes very easily. All right, but if we secrete insulin, and insulin binds to certain receptors and stimulates what are called GLUT4 receptors that come to the surface of the cell, now glucose has a means of getting in. Okay, glucose can now travel into the cell. All right, so glucose is going to go bang. It's going to go in here, and then this gate is going to pop open, and then glucose can travel in, and the cell can break it down for energy. Okay, so that's basically facilitated diffusion. Okay, some kind of gate is allowing the movement, okay, of a certain particle to travel inside the cell. All right, I mean, realistically, that's all it really is. All right, now what's interesting about this, though, is that skeletal muscles, for example, 'cause remember, you guys, a lot of you that are listening to this have heard, they are familiar with insulin, because insulin is a hormone we say is designed to lower blood sugar, okay? And what's interesting is that a skeletal muscle, for example, skeletal muscles, when you're exercising and they're constantly contracting and they're burning energy, they need as much energy as possible. muscles prefer to burn fat, but when you start getting low in your fat reserves, and well, just in general, you need to burn sugar as well as fat. Muscles don't burn pure fat. Okay, so what's interesting is what skeletal muscles can do. They don't need insulin. Just by the contracting mechanisms of the muscle, that stimulates these GLUT4 receptors that come to the surface, and then that will facilitate the movement of glucose across the cell membrane so the muscle can burn it as fuel. Now, why did I just say that? Because a lot of you may either be yourself or know somebody out there that's type 2 diabetes. That's why exercise is a great way to manage a person's blood sugar that's a type 2 diabetic. Okay, because by exercising, we can facilitate blood sugar levels to get down. Okay, plus exercise also helps to control the weight problems that typically coincide with type 2 diabetes as well. Okay, but that's all facilitated diffusion is. All right. The movement of a particle across the cell membrane, and the movement is facilitated by some kind of a gate of sorts. All right, that's all that is. All right, now filtration. Okay, now filtration. What this is, we're talking about filtration. We're talking about basically fluids here. All right, so let me set this up here. Get this as small as possible. Oh, good. Perfect. Aha, perfect. Okay, so let's say this is a hose. All right. Let's say this is a hose. Let's put a couple more. Whoa, that's a big one. Okay. Okay. So with filtration requires some kind of pressure or motive force. All right. Now, when we're talking about filtration here, we're talking about, the movement is being aided by the use of water. All right, movement is being, movement of a particle is happening because of the movement of water. All right. And this is the importance of blood pressure, okay? I mean, you always hear people talk about blood pressure in a negative context, but without blood pressure, we wouldn't have something like filtration taking place. I say that because, when do you hear people talk about hypertension or high blood pressure? When they have a heart problem and they need to take blood pressure medications and so on. All right. So, let's say I was to turn this. Let's say I gave this about half a turn, or I turned this up to about half power. Okay, I turned this hose on. All right, and so you turn this up. Let's say, if you want to visualize this, go to your neighbor's house and punch holes in their garden hose. Because you don't want to do that to your own stuff right now. I'm just kidding. Don't do that, people. I'm just kidding. But so let's say you turn the faucet on here, and water starts to flow through this hose. Okay, water starts to flow through the hose. Okay, when we turn the faucet on, there is pressure that is driving the movement of water throughout this hose. Okay, and within these spaces are holes or pores inside this hose. Okay, so basically, as the water travels by these pores, it's going to filter out. Okay. It's going to filter out, and it's going to continue to filter out as long as there's a driving force like pressure. All right, and there is permeability. There's some kind of pore for the water to travel through. Okay. Now, when you noticed here about these arrows, is that the arrows are the longest, okay, here by the nozzle of the hose here. All right. So the reason why is because these pores right here are closest to the source of the pressure, so there will be a greater rate of filtration. Okay, so the higher the pressure, the higher the rate of filtration. Okay, there's less filtration occurring down, distally out here because because as water flows through the hose, the water is going to be rubbing up against the walls of the hose, and that's going to create some friction, and that's going to slow down the flow of water. Plus, we're getting farther away from our source of pressure, so as a result, there will be a lower rate of filtration, the farther away we get from the faucet of the hose here. All right. Now, this same concept, is applied to the human body. Okay, when we're talking about, the circulatory system, you've got, so let's say we've got a capillary bed here. Okay, so let's say we've got blood flowing down into this capillary bed. Okay, as blood flows into this capillary bed, okay, now you're going to learn, once you get much farther into A&P2, when you learn about the cardiovascular system, arteries are higher pressure vessels, and arteries are classically represented by the color red. Okay, not all arteries are, but typically when you see red, we're talking about a vessel that has blood that's highly oxygenated flowing through it. Arteries are, typically highly oxygenated with blood. Okay, there is one exception, and I'll talk about that when I get to the right when I get there. Okay, and then the blue would represent veins. All right, so arteries, as I just mentioned, arteries are typically known as having more pressure within them. Okay, veins are typically lower pressure vessels, okay, because they're just farther away from the source of the pressure. What's the source of blood pressure in us? I mean, what is the source of the motive force to drive blood flow? It's the heart. Okay, it's the contraction of the ventricles of the heart. Okay. So arteries are closer to, the source of the pressure. All right. So as a result, as blood is flowing through these arteries and there's a higher pressure, on the arterial side of a capillary bed, that's going to favor filtration. Okay, and as a result, now you know that your blood is composed of much more than water. Your blood has ions, electrolytes and ions, same thing. Nutrients like sugars, there are amino acids, there's lipids flowing through the blood. There are gases that are dissolved in the blood or being carried by blood cells, red blood cells. All right, so basically, once we filter this fluid, the plasma, the water out of our blood into the tissues, this is how we carry the nourishment or the nutrients for the cells. This is how we get the sugars and the gases, the oxygen, and all the other nutrients into the cells. Okay? And then as blood continues to flow through this capillary bed, okay, the filtration rates are going to go down because, remember, of all the friction I just mentioned and the distance from the source of the pressure. Okay, so as a result, remember, there's going to be water in this environment anyway. So because the pressure in the vessel is getting lower, that's going to favor a process called reabsorption, which we'll talk about when we get to the cardiovascular system. Okay, but so that's basically how filtration works. And filtration is necessary because, as I mentioned, this is how we deliver essential gases and nutrients from our blood to our tissues. Okay, this is how we create urine as blood flows through kidneys. Okay, because as the blood flows through the kidneys, the same thing. We filter our blood by driving fluids out of capillaries, and then we process that through cellular structures called nephrons in the kidneys, and then we form urine as a result. Okay, so that's filtration. Okay, using water as a transport medium, but we need pressure to we need pressure to drive the movement of water and drive the transport of these substances as well. And that's what happens when people have low blood pressure. People that have a significant drop in blood pressure, okay, you'll have a lower rate of filtration. Okay, so you'll have a harder time delivering nutrients and oxygen to your tissues. All right. Vice versa, if you have hypertension, you're running the risk of edema. Okay, because the higher the pressure, let's say I went from half power and I turned this thing up to, full power. Okay, and I had water blasting through here at a higher rate, more water is going to be pushed or filtered out of the hose. Okay, same thing with us. Okay, the higher the rate of filtration, the more, we're going to disrupt the fluid cycle of our body, and we could develop edema as a result of this. That's why one of the many reasons why keeping blood pressure under control is essential. Okay, but like I said, blood pressure is an essential part of life. Like I said, just don't always think of it in a negative context. All right, so that's filtration. Now next, let's talk about probably one of the most confusing topics that tends to get people, osmosis. Okay. Osmosis tends to be confusing just because people tend to look at it, just because of how people look at it. All right. So let's draw this on here. All right. Let's say we've got a membrane here. Okay, we've got a membrane there. All right. Now, this is what's going to be our, I'm going to abbreviate this, selectively permeable membrane. All right. And then we'll call this side A, we'll call this side B. Okay, we'll call that side A, we'll call that side B. Okay. Now, excuse me. So now, first, we have to let's kind of zoom in and take a look at this membrane. All right, let's kind of zoom in and take a look at that membrane. Okay, so as we're taking a look at this membrane here, all right, we're going to notice there are going to be some pores on here. That's a terrible picture. It's the reason why I study biology, not art, folks. I'm sure you're getting the gist of that as you watch these. Okay. So, let's say, for the sake of discussion, that this is about, we'll say this is a water molecule. We'll say that's a water molecule, that's a water molecule, that's water, water, water. Okay. And let's say these are, oh, I don't know, let's just say, I don't know, we'll say these are proteins. Okay. What do you notice about the size of these pores? Okay, what do you notice about the size of these pores? Okay, you'll notice that for starters, again, as we mentioned earlier, in order for osmosis to take place, there has to be a selectively permeable membrane. The same concept as diffusion as well. There has to be a selectively permeable membrane. Except in this situation, when we're talking about osmosis, the membrane is selective to water. Okay, the membrane is selective to water. Okay, so you know, this membrane is small enough to allow water to pass through it, but it won't allow proteins to pass through because the proteins are too large. Okay, so that's number one. Okay, that's number one. There has to be permeability. Okay, now number two, we have to look at the concentration of water. Okay, we have to take a look at the concentration of water. All right, when we're talking about this, what we need to understand is that this is where people tend to get mixed up. Okay, so let's take a look at these proteins here. Okay, now remember when we talked about water in chapter 2 and that really basic chemistry stuff we were talking about. Now remember we said that water is polar, and water does not chemically but physically interact with other polar substances dissolved in it. Okay, so let's say here's the protein. Okay, and remember we said that the polar sides of the water, okay, will interact with whatever the substance is that's dissolved in it. Okay, so here's where people tend to get mixed up with this. So we just got done talking about diffusion. Okay, we just got done talking about diffusion. Okay, now, being that we just got done talking about diffusion, you guys are going to look at this. Now let me ask you right now, what direction is water going to travel? If let's say this membrane is permeable, it's ready to allow water to pass through it. How is it what direction is it going to travel? Think about it for a sec. Excuse me. Okay. Most of you probably said that water is going to travel in this direction. Okay. If you did, You're wrong. Okay, if you did, you're wrong. Okay, because this is the mistake people make. They look at the solute concentration and they think the solutes are going to be moving. Okay, remember, I just said that this membrane is only permeable to water. Okay, so whatever solutes, or I'll just keep sticking with particles, whatever particles, the particles that cannot freely pass through the membrane, okay, we call those osmotically active. Okay, we call those osmotically active. Okay, so what we're saying here is that these can't freely move across the cell membrane, but because of this right here, all right, they can attract or draw water to one side or the other. Okay, so there being that there are more osmotically active particles on this side, on side B versus side A, that's going to draw the movement of water in that direction. Okay, that's going to draw the movement, the attraction of water in that direction. Okay, and you know what's going to happen is then that's going to, you know, and again, just like diffusion, this water is going to be drawn in this direction until, you know, again, the forces equilibrate. Okay, until the concentration of water is going to be equal on, you know, on side A and side B. Okay, that's, you know, again, this is a very basic explanation of this because I didn't go into molarity, molality, and all that stuff, but that's the basics of how osmosis works. Okay, you know, how do these osmotically active particles affect the movement of the transport of water across the cell membrane? Okay, and again, the more osmotically active particles on one side versus the other, okay, you know, that will influence osmosis. Okay, so you need to focus on the concentration of the particles, but you also need to focus on how that's going to affect the movement of water. All right. Now, when we talk about a topic like this, you know, this is an important topic to understand because you're going to, I mean, as you're going to go through every single chapter of this book, and we're talking about all these topics, a big part of our, you know, our cells staying healthy is maintaining water balance, maintaining what's called the fluid cycle. Okay. And basically, a big part of this osmosis topic that we're talking about is something called tonicity. Okay, when we're talking about tonicity, what that is, that's what we're talking about is the effect of a solution on the osmotic movement of water. Okay, the effect of a solution on the osmotic movement of water. Okay, so let's say, for example, so let's say I've got a beaker here. Okay, you guys are going to be doing this. I know that some of you, you know, human biology students, you know, taking the prerequisite course of this are going to be doing this. All right. So let's say I take a cell and let's say I take a red blood cell and I submerge it in water. All right. All right. Let's say I take that red blood cell and I submerge it in water. All right. And on the inside, there are 300. Now the units I'm just going to write the unit up here. I'm just going to write the units up here. You know, 300 milliosmoles of water. Okay, so those are the units I'm using, milliosmoles of water. When I'm saying milliosmoles, I'm just talking about, okay, what are, you know, how are these particles affecting osmosis? Okay, so let's say there are 300 milliosmoles of sodium on the inside of the cell, and let's say the concentration of sodium in the water surrounding it is 300. Will there be a net movement of water into and out of the cell? Because remember, there's always going to be movement taking place. There's always going to be movement in, movement out, okay? But in this situation, there will be no net movement, okay? Due to the equal amount or equilibrium of sodium on the inside versus the outside, okay? So as a result, this cell will be able to maintain its shape. Okay, and that's extremely critical that cells maintain their shape, because you saw the picture earlier I showed you the cell that had the nucleus, that had the you know, the endoplasmic reticulum and the Golgi apparatus, the lysosomes and all that. All right, because one thing you have to remember about cells is that all these working parts of the cell, you know, a big part of their functionality is the distance between the parts. Okay, is the distance between the parts. All right. So what's going to happen then is, let's say we become hypertonic. Okay, we become hypertonic. So let's say I take this beaker of water and I plunk a red blood cell in there, and again, the internal concentration is 300 milliosmoles. Okay, 300. Now, let's say that the concentration outside of the cell, this would be the equivalent of the extracellular fluid, is 600 milliosmoles. What direction is water going to travel? What's going to happen to the cell? There will be osmosis taking place, but what's going to happen? Okay, remember, sodium is an ion. It's positively charged, right? So remember, how water interacts with charged or polar particles. All right. So what we're saying here, just by looking at the numbers, we're saying that there are 300 more osmotically active particles in the extracellular fluid versus the intracellular fluid. So what we're saying here is that there are more particles that are going to affect osmosis. So being that there are more sodium ions on the outside, that means we're going to draw more water out of the cell. Okay, we're going to draw more water out of the cell. There's going to be an imbalance. There's not going to be an equal amount of sodium moving in and out of the cell anymore. There's going to be more water drawn out. Okay, and what the cell is going to do then is it's going to shrivel up. Okay, it's going to shrivel up. All right, and then what's going to happen then is, if this cell shrivels up, all the working parts of the cell are going to get scrunched together. Okay, and if these organelles get scrunched together, all right, they're not going to function properly. Okay, they're not going to function properly at all. Okay, the chemical reactions that are driven within these cells aren't going to operate properly, because there needs to be a certain amount of space and timing that between these organelles that to allow them to function properly. All right. So if this cell shrivels and gets compressed, the organelles are going to squeeze. The organelles are going to squeeze together. All right. And if we don't correct this, the cell's eventually going to die. Okay, because it's not going to be able to undergo its number one function, synthesizing proteins. All right. So if we become hypertonic, okay, that's bad. All right. The opposite of hypertonic, this is what we would call isotonic. Okay, the opposite of hypertonic would be hypotonic. Okay, so let's do this again. All right, we got our red blood cell, and the red blood cell has 300. And now let's say there are only 100 sodium ions in the extracellular fluid here. Okay, now we create the exact opposite of this situation here. This is what we would call hypotonicity, or being hypotonic. All right, so what's going on here now is, if I have more osmotically active particles on the inside of the cell versus the outside, that's going to draw water into the cell. All right, that's going to draw water into the cell. And then what's going to happen here then is the cell is going to get bigger. So let's say here's what the cells should normally look like in size. Now all of a sudden it's going to swell and get bigger. Okay, it's not going to burst. The cells aren't balloons that are going to pop when you fill them with too much air. Okay, what's going to happen is, the exact opposite of this situation as described over here. Okay, the all the working parts of the organelles of the cell are going to get shoved farther away from one another. Okay, and it's going to take longer for the cell to do its job. It's going to take longer for it to crank out the essential proteins that are required to keep it alive. Okay, and if there's a greater distance in between these organelles, and it takes it a lot longer to make its products to keep it alive, it's also going to take longer for carbon dioxide to diffuse out as well. All right, it's going to take longer for oxygen to get into the working parts of the cell. So in general, you can figure out what's going to happen here. The cell is also going to die. all right, if we don't correct this problem as well. All right. Now, like I said, I mean, almost every single book out there likes to say the cells are going to burst. They don't do that. All right. They'll break apart after they die, but they're, like I said, cells aren't balloons. All right. So let's talk about some real life scenarios when we would see this happening. All right. So, for example, example of becoming hypertonic would be if you're dehydrated. Okay, when you're dehydrated, okay, let's say, one of the most common causes of dehydration is sweat. Okay, let's say you're out on a midsummer day doing yard work, or you're exercising, and you're not properly filling yourself with the water you need. Okay, when you sweat, okay, you lose water and salts. Okay. Now, you're not going to sweat out the same ratio of water and salts. You're going to lose more water than you are salts. Okay. And the major salt that you find in the extracellular fluid, the major electrolyte, is sodium. Okay, sodium. Okay, when we're saying 300 milliosmoles here, we're talking about we're always maintaining 300 milliosmoles of sodium in extracellular fluid. Okay, so if we're sweating out more water than sodium, what's going to happen then is as we lose fluids, okay, our body fluids are going to become more concentrated with sodium. All right, so if our extracellular fluid becomes more concentrated with sodium, that's going to make us hypertonic, and then that's going to force water to be drawn out of the cells and into the tissue spaces, into the blood, to try to keep our blood volume and pressure back up. And again, if we don't correct this is going to kill us, okay? And the cells that are really going to be affected by this are neurons, okay? Because nerves, you're going to learn as we go later on, nerve cells and neurons are really finicky when it comes to changes in their environment or physical changes to themselves, okay? If neurons start to have their shape altered, they just may not function altogether. That's why when people are severely dehydrated, you're going to see neurological effects taking place pretty quickly. All right. And the opposite of this would be being hypotonic. This is why they say don't drink distilled water. Okay, distilled water is pure water. Okay, and I kind of live the general rule of life that's good to live by. If you can put it in your car, don't put it in yourself. All right. But distilled water is pure water. So if you sit there and pump yourself full of distilled water because you're really thirsty and you don't know any better, okay, you're going to dilute your extracellular fluid, and then as a result, you're going to see this taking place. Okay, and that's bad as well. Okay, so don't drink distilled water. All right, drink regular water. All right, so we work hard on a day-to-day basis to make sure that we are staying isotonic, to make sure there's no real major net movement of water into and out of the cell, that we're always at an equilibrium between these osmotically active particles. Okay, and that's osmosis, the movement of water across the cell membrane. Okay, so again, if you have questions on this, let me know, because this is important for you guys to understand. Because, this is going to help you understand edema. This is going to help you understand water regulation in the body. This is going to help you understand a lot of important topics. Okay. So next, what I want to talk about is active transport. Okay. And when talking about active transport, there are, again, as I mentioned before, there are different modalities of active transport that occur, but I'm going to keep it basic. Okay, remember when we're saying active transport, this requires energy. Okay, active transport requires energy to to undergo this, because remember, active transport is a movement at an uphill gradient. We're working against a concentration gradient. Okay, we're moving from an area of low concentration to high concentration. Okay, this takes energy. All right, this takes energy to do this. So, let's say we've got a cell, and on the inside of this cell, we've got, 10 sodium ions, and on the outside, we've got 145 sodium ions. And let's say on the inside of the cell, I've got 145 potassium ions and 10 on the outside. Okay. during active transport, this is a big part of our daily energy expenditure right here. Just undergoing this active transport process to make sure that nerve and muscle cells maintain a normal resting electric charge. Because I'll talk about this more specifically later on. Something called membrane potential. Membrane potential is an electrical charge that's created basically by establishing a certain concentration of sodium and potassium across the cell membrane. Okay, and in order to maintain a normal resting membrane potential, or resting electrical charge, we'll call it, okay, there needs to be the right amount of sodium across the cell membrane and the right amount of potassium across the cell membrane. Okay, every now and then potassium is going to leak out. Every now and then potassium, we're going to have sodium move in, and as a result, we need to reset this concentration. Okay, so then what's going to happen is we're going to actively pump sodium out and potassium back in. Okay, and we do this by using what's called the sodium potassium ATPase, or the sodium potassium pump. Okay, remember when you see the letters ASE at the end of a word, that represents an enzyme. Okay, so we have an enzyme breaking apart ATP to release energy. All right. So basically, we'll typically move 3 sodium ions out, 2 potassium ions in. Okay, but in order to do this, you can see the numbers that we've established here. We're working against the concentration gradient. Okay, and again, this is going to require the use of ATP. All right, this is going to require the use of ATP. Okay, so as a result, if we're pumping sodium out, we need to burn energy. Potassium out, we're burning energy as well. The nice thing about this is that this is that these move simultaneously. As sodium is moving one direction, potassium is moving the other. Okay, but that's essentially all active transport is moving against the concentration gradient. Okay, and I'm going to talk more about this later on when I talk about this, when I talk about skeletal muscle contractions, and we talk about pumping calcium back to where it belongs when we're trying to reset a muscle back to a resting state. When I start talking about nervous tissue, we're going to come back to this concept much more in depth when I talk about the maintenance of a resting membrane potential. Okay, but that's all, that's basically all active transport is. Like I said, I know I'm not going terribly in depth with this right now. I'm going to go in depth with it more later on when I get closer to where I can really directly apply this to certain tissues of the body. Okay. Now, lastly, talked a little bit about bulk transport, and there are 2 major types of bulk transport that happen in the body, and that's what's called exocytosis and endocytosis. Okay, I always miss one little part there. Okay, exocytosis and endocytosis. Okay, so we're talking about exocytosis. What I'm saying here is, what I'm talking about here, think of exo as out, external, endo as internal, within. So we're moving a large amount of something out of the cell, or a large amount of something into the cell. Okay, and basically with exocytosis, a very common example of that, let's say we're talking about the release of a neurotransmitter. Let's say this is the very end of a nerve cell. It's called a synaptic terminal. Okay, there are these little vesicles inside the synaptic terminal here. Okay, and inside these vesicles are neurotransmitters. Okay, neurotransmitters are chemical messages that nerve cells release for the purpose of communication. All right. Now, when the time is right, when this nerve is stimulated and it generates an electrical signal, an action potential, what that's going to happen is that's going to reach this little synaptic terminal here, and that's going to stimulate bulk transport, exocytosis. Because what's going to happen then are these little vesicles, what are called synaptic vesicles to be specific. These vesicles what they're going to do is they're going to fuse with the plasma membrane. Okay, so let's say we've got a cell membrane right here, and we've got a synaptic vesicle. Okay, what's going to happen is it's going to fuse with the plasma membrane. It's going to look like this, okay? And then basically what happens then are all those neurotransmitters that are now located inside of that vesicle are going to be, are going to be dumped out of that cell, okay? The release of a neurotransmitter, and that requires, that's an energy taxing process right there. All right, so that's an example of exocytosis, bulk transport. We're moving a lot of neurotransmitters at once out of the cell, hence the term bulk transport. Okay, and a commonly used example of the opposite of this would be something called phagocytosis. Okay. This in medical terminology, think of this as, eating, consuming, appetite. Okay, there are certain cells of the body that eat things, that consume certain materials and products that are found within us. Okay, so we'll use the example of a phagocyte. Okay, if you remember, phago, eating, cyte, cell. Okay. Now, a very common type of a phagocyte would be what's called a macrophage. These are phagocytes that are found in a wide variety of tissues in the body. But sometimes we need to, these cells need to, keep the tissues clean. Okay, whenever we cut or damage a tissue, whenever there's an infection and we have to fight it off, whenever there's just a foreign invader in general, okay, there's going to be tissue damage of some sort, and we can't start the healing process without cleaning the mess first. If you have a tornado blow your house to bits and smithereens, okay, you cannot rebuild the house on the foundation until you clean the rubble off of the foundation. Okay, so what a phagocyte's going to do then is it's going to approach, let's say this is a small object, all right, so the phagocyte is going to approach this, and it's going to act in an amoeboid-like fashion, it's going to act like an amoeba. You guys remember amoebas, these kind of big cells that kind of squirm and slither their way around when they have to move. Okay, so what the cell is going to do, it's going to get close to this foreign object or this damaged tissue or whatever it is it's going to consume, and what it's going to do is it's going to create a little inward fold of its cell membrane, or an invagination of its plasma membrane. Okay, and then as it kind of starts to create this fold around the object, it's eventually going to create this vesicle or this sac. Okay, from its membrane, and then this sac is going to then travel its way into the phagocyte, and then there are basically going to be these intracellular organelles that are going to diffuse with this vesicle, dump out digestive enzymes, and break apart whatever it was that it just consumed. Okay, so phagocytosis, endocytosis, exocytosis, examples of bulk transport. And again, you're going to hear about these later. When I get to the nervous system, I'll talk much more about. I'll talk a lot more about. About this synaptic vesicles and exocytosis, and when we get to the immune system, I'll talk a lot more about phagocytosis and phagocytes. Okay, so again, folks, I can't say this enough. If you have questions, contact me, and well, good luck in your studies.